One of Morgan's grad students looked at the data. One of the people involved in the recombination frequency we talked about in lecture E. Looked at the data from the recombination experiments and noticed that the recombination frequency is different between different genes, different phenotypes. For some, between these two, it would be quite a small number. Others, it's approaching 50%. Not quite 50% because they're still linked genes. But he was able to figure out and theorize and figured out an explanation for this. The closer two genes are on the chromosome, the chances of the crossing over between them is going to be much, much less than two genes that are further apart. Further apart means that there's a greater chance of a crossing over, a chiasmata forming between the two genes. Closer they are, minimal chance of that occurring. So Sturtevant started to put together what we now refer to as a linkage map, what he referred to as a linkage map. Remember back when you took the SATs, the ACTs, there's always those stupid questions I hate. Bob wants to sit next to Jill, but Jill needs to sit 2 seats away from Tom. Tom can only sit next to Jim, and Jim has to sit at least 4 seats away from Bob, and blah blah blah. What was the seating arrangement? Remember those stupid questions? Hate those stupid questions. But that's what this linkage map is. Going through and calculating probabilities. The probabilities of a recombination event occurring between two close versus two further away genes. Each percentage was referred to as one map unit, or a centimorgan. Centimorgan is not a physical distance, it's a recombination distance. The higher the centimorgans, the greater the chance of a recombination occurring between the two genes. So, the recombination frequency between B and VG was 17%, thus 17 centimorgans. Further experiments showed that the difference between the recombination frequency between B and CN, CN's the cinnamon eyes, was 9%, and the recombination between CN and VG was 9.5. Nine plus 9.5, 18.5. It's not exactly 17%, but you got to remember you have to also take into account we're dealing with, nature. We're dealing with living organisms, so there's going to be a little bit of play there. But 17, 18-ish% is roughly the same thing when you're looking here. It's not going to be perfect, but it's pretty close. So the relative differences between the genes could be additive, is what they figured out. Further apart they are, the closer that will come to 50%. If it ever becomes a recombination that's 50%, Mendel kicks in, and it turns out those two genes are on separate chromosomes. So Sturtevant went through and supervised, did many of them himself, many different combinations of fruit fly breeding experiments with mutant phenotypes, or mutant types that seem to be linked. And then he laid it out. What was the seeding arrangement? What they were able to, Sturtevant was able to calculate and figure out is the first genetic map. He figured out the loci. They refer to this as a cytogenetic map, a cytocell genetic map. On the chromosome, at one end, will be the gene for, or the mutation for short antenna. 48.5 centimorgans away, black body. What is that? 9 away, cinnabar. 9.5 away, vestigial wings. And then you go whatever this is, I'm not going to do the math, to get brown. And he just kept going on. So he figured out the seeding arrangement. Short, black-bodied, cinnabar, vestigial wings, brown-eyed, so on and so on and so on. All from looking at just the numbers.